There's a skull in a drawer at the Yunnan Institute of Cultural Relics. Her mitochondrial DNA was finally sequenced, and it landed on a branch of the human family tree that had never been seen before in any person, ancient or modern, anywhere on Earth. She is 14,000 years old. Her thigh bone matches a Homo erectus from 1.5 million years ago. She sat in storage for 19 years before any scientist was permitted to study her. I want to be careful here, not in the sense of hedging facts, but in the sense of distinguishing between what the science actually says and what people claim it says. There is a significant difference. So let me start with what we know for certain. In 1989, Chinese archaeologists working a cave called Maludong, the Red Deer Cave, near Mengzi City in Yunnan province, recovered the partial remains of at least 3 individuals. They found charcoal, burnt clay, the bones of a giant extinct red deer, and a fragment of a human skull cap, the frontal bone, and the paired parietals. They cataloged it, they put it on a shelf, and for the next 19 years, no formal scientific study of those bones was conducted. The fossils were not lost. They were shelved. There was a second discovery 10 years earlier. In 1979, a petroleum geologist named Li Changqing was surveying caves near Dae village in Longlin County, Guangxi province, and he noticed a sediment block embedded with human bone. He shipped the block to Kunming. A mandible and some body bones were extracted from the rock. The skull was still locked inside. The block was placed in storage. It would remain sealed in that rock, untouched, for 30 years. In 2008, an Australian paleoanthropologist named Darren Curnoe began collaborating with Xueping at the Yunnan Institute. They were re-evaluating East Asian fossil collections, and they found these forgotten boxes. Curnoe and G built a team, 6 Chinese institutions and 5 Australian institutions. In 2009, the Longlin block was finally cracked open, and the skull was reconstructed. In 2012, in the journal PLOS ONE, Curnoe and G published the first formal scientific description of what these bones actually were. And the description was a problem. The skull did not match. The frontal lobe was unusually long, measuring 86 millimeters, unusually broad, 121 millimeters, unusually tall, 92 millimeters. Those proportions resembled early Upper Paleolithic European Homo sapiens, far more than they resembled any recent East Asian. The cranial bones were thick, the brow ridge was prominent and archaic. The brain capacity was smaller than modern. The forehead curved in a shape that did not appear anywhere else in the fossil record. The eye sockets flared unusually wide. The cheekbones flattened and projected in a combination that had never been documented in any other specimen, archaic or modern. Curnoe and G refused to assign her to a known species. They described her as an unusual mosaic of archaic and modern features, and they floated the possibility of an unknown population. Then, in December 2015, the same team published a second paper about a femur fragment from Maludong, the thigh bone of one of these individuals. The femur did not match modern humans either. It matched KNMERR 1481, a Homo erectus specimen recovered in Kenya, dated to roughly 1.89 million years ago. A thigh bone of an archaic hominin from 1.5 million years before agriculture was beating against the same cave floor where humans, somewhere on the planet at that exact moment, were already domesticating wheat. She is the most recent archaic feature human ever found anywhere in the global fossil record, by tens of thousands of years. Think about what that sentence actually means. By 14,000 years ago, humans had crossed the Bering Land Bridge into the Americas. Humans had painted the caves at Lascaux and Altamira generations earlier. The last Neanderthals had been gone from Europe for more than 20,000 years. The Denisovans had vanished. Homo erectus, the species whose femur this skeleton's thigh bone resembles, had been extinct for roughly 110,000 years in Asia by the most generous estimates. And yet in a cave in Yunnan, on a date when the climate was already warming toward the Holocene, when wild grains were already being gathered in the Levant, somebody with the thigh bone of a million-year-old hominin and a forehead that exists nowhere else in the fossil record was sitting next to a fire eating a giant red deer. Curnoe and G proposed she might be a hybrid, a modern human population that had interbred with an unidentified archaic species. Possibly a Denisovan, possibly something we have not yet named. The peer review pushed back. National Geographic quoted critics arguing the mosaic features fell within late Pleistocene East Asian variation. Curnoe responded in print that the femur, in particular, was harder to dismiss. The paper stood. And then nothing happened for 10 years. Between the morphology paper in 2012 and the next major publication, an entire decade passed. The skull sat in Kunming. The femur sat in Kunming. The question sat in Kunming. The world's most morphologically anomalous late Pleistocene human, a fossil that the lead authors had openly suggested might be a hybrid with an unknown archaic species, generated a decade of scientific silence. No follow-up morphometrics, no comparative analysis with the Denisova samples emerging from Siberia. No DNA. Nothing. Until July 2022. In that month, a team led by the geneticist Bing Su at the Kunming Institute of Zoology, with first author Xueping Zhang, published in the journal Current Biology a paper titled A Late Pleistocene Human Genome from Southwest China. They had finally extracted ancient DNA from MLDG 1704. They recovered nearly the complete mitochondrial genome and roughly 3.3% of the nuclear genome. And here is exactly what they found, because the precision matters. Her nuclear DNA places her firmly inside Homo sapiens. She is not a Neanderthal, she is not a Denisovan, she is not a separate species. That is what the science actually says. But her mitochondrial DNA, the line passed unbroken from mother to daughter, sits on a previously unsequenced basal branch of haplogroup M9. A branch that had never been recovered in any human being, living or dead, before MLDG 1704. A matrilineal line that left no living descendants. It went extinct. Whatever population she belonged to, that lineage ended. The branch is real. It is on GenBank. You can download it. And before 2022, no one on Earth knew it had ever existed. That is what the phrase doesn't match any known human lineage refers to. Not species, not nuclear, the matrilineal branch. The previously invisible branch of the human story that was hiding in a Chinese drawer for 33 years. To make this concrete, haplogroup M9 is not new. It is one of the deep East Asian mitochondrial lineages sampled in living populations from Tibet to southern China. But the branch she sits on splits off near the very base of M9 before any of the sublineages that survive in modern people. The closest relative they could find for her was the Tian Yuan individual, a 40,000-year-old skeleton recovered from a cave near Beijing. 40,000 years and over 1,000 miles of separation. And the closest thing to her on the entire mitochondrial tree is a much older woman from a different province. Everyone else on her branch is gone. Zhang's team went further. They argued that her genetic profile sat almost equidistant between ancient Northeast Asians and the founding populations of Native America. They argued she was evidence for the Southern Route hypothesis, that some of the ancestors who crossed Beringia and seeded the Americas did not come from Siberia at all, but from somewhere closer to where she was buried, in southern East Asia. The headlines went worldwide. CNN ran it. The South China Morning Post ran it. A 14,000-year-old woman from Yunnan was suddenly genetically closer, by some measures, to the first humans who walked into the Americas than to anyone living in China today. And then in 2024, the story broke a second time. Independent geneticists published a formal critique in Current Biology. They reanalyzed Zheng's published sequences and reported extreme error rates, abnormal error distribution patterns, and evidence of contamination by modern human DNA. They could not replicate the first American's link. The basal M9 identification, that piece, held up. But the headline, the part that put her on every news site on the planet, the part about Beringia and the southern route, could not be reproduced from the data as published. The team responded with an addendum. The mystery did not dissolve, it deepened. There is one more detail worth pausing on. The contamination critique was not a fringe blog. It was published in the same journal, Current Biology, that had published the original genome paper. The reviewers who flagged the errors were independent ancient DNA specialists. Their language was technical, but their meaning was direct. The error rates in the published sequences were too high. The distribution of those errors was inconsistent with degraded ancient DNA and more consistent with modern human contamination. And the headline phylogenetic claims could not be reproduced from the data on file. That is not a small disagreement. That is a peer-reviewed challenge to the most globally publicized finding ever made about this fossil, and it took 2 years to surface. So now stack the silences against each other. The Longlin block sat sealed in rock for 30 years before anyone opened it. The Maludong fossils sat in storage for 19 years before anyone studied them. 10 more years passed between the morphology paper and the DNA paper. 2 more years passed before the contamination was flagged. From the moment a petroleum geologist noticed bone in a sediment block in 1979 to the moment we had a verified, replicable, published account of who this woman actually was, 45 years went by, and we still do not have one. The fossil is in Kunming. The catalog number is MLDG 1704. The DNA is on GenBank. The morphology is described in PLOS ONE. The femur is described in PLOS ONE. The mitochondrial branch is in Current Biology. The contamination critique is in Current Biology. Every piece of this story is technically public. The question is not what we do not know about her. The question is why it took so long for anyone to be allowed to ask. 19 years on a shelf, 30 years inside a rock, a decade between papers, a 2-year gap before the contamination was named. The bones never moved, the drawer never moved, the institution never moved. What moved, slowly and reluctantly, and only after the right people insisted, was the permission to look. She is anatomically modern. She is genetically modern in her nuclear DNA. But her face is wrong. Her thigh is wrong. Her mitochondrial line is invisible everywhere else on Earth. And whoever her mother's mother's mother was, that woman walked out of a population that left no other trace. None. Anywhere. In any cave, in any genome, in any living person on this planet. The silence is the real mystery. It always was. 14,000 years of it inside the bone, and 45 more years of it inside the institution, and somewhere in between, a branch of the human family tree that we are still not entirely sure we are allowed to study. She is in a drawer in Kunming, catalog number MLDG 1704, and the science has not finished with her yet.